Good evening, everybody. Welcome to your first online anatomy class. Tonight we're going to be looking at the muscular system. It's going to be chapter 20 in your textbook. So let's get right into it. Before we get started, it's important to understand a couple prefixes that we're going to commonly see. The first one that you'll notice is going to be myo. Myo is going to be Greek for the term muscle. So we're going to constantly see myo in front of a lot of the terms we look at. And same thing with sarco. Sarco is also another one. And this is going to be flesh in Latin. It's important to understand that complex life is going to be built around the ability to move. So an organism is going to have to be able to respond to its environment. So it's going to need a nervous system to tell the muscles of the body how to move, how to respond. And interesting enough, life that no longer needs the ability to move, like certain sea sponges, are going to find a rock and plant down, and they're just going to get rid of their nervous system because it's not important to really move around the environment anymore. And the person, or the physiologist that discovered how muscles physiologically contract, his name was Hugh Huxley, and that wasn't until 1969. So relatively new as far as some of the other physiological activities we've looked at so far. As far as what muscles are responsible for doing, the big one's going to be movement, obviously. So as these muscle cells contract, they're going to pull on the skeleton, the attachment points on the bones, that move across the joints, and it produces movement. Another type of movement they're able to produce is movement of the blood and lymph within the vessels of the body. So here we can see a vein, and within the vein, the blood has to be able to get pushed back up against gravity to get back to the heart. So to assist moving against the gravitational pull, we're going to have muscles around. So here we have one-way valves that help prevent the blood from pooling downward. But we're also going to have muscles that surround these vessels. And every time they contract, they're going to push on these walls, squeeze the blood up higher to the heart. And then when they relax, the valves close back down and the blood is now moved further up the body. So the constant contraction and relaxation of these muscles surrounding the veins and the lymph vessels is going to be what increases circulation. Sometimes you might see certain people at weddings, choirs, or especially the military, if they stand at attention for too long and they're not moving their legs and contracting those muscles, the blood's not going to get to their brain and they're going to end up passing out or fainting because of a lack of circulation. So it's always important to keep your muscles contracting when you're standing for a long period of time. Another physiological characteristic of muscle is it's important for postural maintenance. So the ability for me to sit here upright, face forward, head up. I'm going to have a constant tone in my skeletal muscles. Here we can see someone that suffered a spinal cord injury, so their nervous system is no longer able to send a signal to their muscles to tell them to contract or maintain posture. So for this person to maintain a standing position, he's going to need physical assistance through other measures because his body can't develop them themselves. And we can also produce heat through our muscular system. Anytime we have chemical reactions in the body, it's going to generate heat. So as muscles use up all that ATP that we learned about, the breaking up of that ATP molecule is going to release energy and it's going to create heat. And this is what we call thermogenesis. Thermo meaning temperature, genesis meaning creation. So here we can see an infrared picture of somebody. The darker red and green areas are going to be a higher temperature. Here it says 96.4 Fahrenheit. Whereas the blue or purple areas are a colder temperature, like 70 degrees. So if we looked at a picture like this, we could surmise that he was using his arm, shoulder, and chest muscles more than the rest of his body. And this is also a reason why we shiver when we're cold. When our body temperature gets to a certain point too low, the brain's going to take over the skeletal system and cause a spontaneous quivering and shivering of the muscle tissue to help try to generate that heat. And that's going to raise the body temperature as those muscles shiver. Some specific properties of muscle cells, there's going to be 4 that we look at. The first one is muscles are able to be excited. And when we say excited, we mean they're going to be able to respond to signals from the nervous system. So here we can see we have 2 neurons. This upper motor neuron extends all the way from the brain, goes down the spinal cord, and it's going to connect or synapse to a lower motor neuron, which eventually targets its specific skeletal muscle that it's going to send a signal to. And it's only going to be nerves and muscle cells that have this property. We'll look more closely at action potentials, but basically it's going to be a flow of ions, so an electrical current that is going to be able to stimulate these muscles. So here we can see an action potential firing. So we thought of voluntary, I want to move my, in this case we'll say this is the biceps, I wanted to move my biceps, I fire that action potential, it goes all the way down to the muscle, and then as the muscle gets that signal, it's going to become excited and those muscle cells are going to contract, and when we say contract, they're going to physically shorten, and that's what's going to generate force and cause contraction of the muscle. So again, we can look at that. We fire an action potential, goes to the muscle responds, action potential is finished, so the muscle goes back to relaxing. Muscles are also able to extend, so we call this the property of extensibility. So even since muscles can contract, they also have to be able to relax and extend themselves. So here our elbow is flexed, and then as we stretch it, we're going to extend that elbow, and the muscle fibers are going to elongate to accommodate that movement. Obviously, this is important to produce full range of motion. At the same time, muscles can also extend while generating force. So if we're holding a weight in our hand like here, as we lower our forearm, this muscle is still generating a force. If it wasn't, we would just completely drop that weight without any control. So this ability to generate force while extending is what allows us to create smooth movements while holding a load or weight in our hands. And lastly, we have elasticity. So this is going to be the muscle's ability to return to its original shape. So it can contract, it can stretch, but we want it to go back to its original shape when we're done using it. Looking at the anatomy of how muscles are set up, they're going to be kind of stacked layers on top of layers. So we're going to start small and then work our way out larger. So first, we'll start with a muscle fiber, and a muscle fiber is just another term for a muscle cell. So I know when we did the skeletal system, we kind of saw cells as this kind of round shape here, and with our nucleus and all the other organelles. But as I said, within the body, cells are going to have all different shapes and sizes. So in the case of a muscle cell, they're going to be much, much longer. So in the case of a biceps, it's going to go all the way from the shoulder all the way down to the elbow. So they can be as long as the joint or the tissue themselves. So here we're looking at a muscle cell then. And then around each muscle cell, we're going to have a connective tissue sheath that protects it and serves a function. So the connective tissue that surrounds individual muscle cells is going to call endomysium. Again, if you remember, the prefix endo means innermost or inner. So endomysium surrounds each individual muscle cell. From here, we're going to build out and we're going to stack a bunch of muscle cells together. And this bundle of muscle fibers is going to be called a fascicle. And each fascicle is going to be surrounded by another sheath of connective tissue. This time, it's going to be called perimysium. And perimysium is going to be the connective tissue that surrounds the fascicles. From here, we're going to stack a bunch of different fascicles together. And as all these fascicles here, so you can see this is one fascicle, 2, 3, 4, 5, 6. So many fascicles come together, and this is going to make up the bulk of the entire muscle. So a group of fascicles is what we normally just call the muscle themselves. So this is the whole thing, everything together. And around the muscle, or the groups of fascicles, we have a final layer of connective tissue that surrounds the entire muscle, and that's going to be called epimysium. Epi meaning on top. So here we can see our epimysium at the very top or outside of the muscle fibers. So again, if we look at a cross section then, this is our muscle fiber, we're looking straight at it now, and around these muscle fibers, we have our endomysium. We're going to get a bunch of muscle cells, put them together, and now these, let's say this group of 7 muscle cells has made up the entire fascicle. Around just one fascicle, we have our perimysium. And then we're going to have a bunch of fascicles together, and now we have our entire muscle. And around this entire muscle, we have the epimysium. So it's just layers stacked on layers. So now we're going to just zoom in and concentrate on the individual muscle cell themselves. So we learned about the different organelle names previously, and these organelles are going to exist in muscles, but we're going to have different terms for them. So in the case of a cell membrane, we're going to call it the sarcolemma. So this is our muscle cell here, and the outside covering the cell membrane is what we call the sarcolemma. The cytoplasm inside the muscle cell, so all the fluid on the inner aspect of the sarcolemma, is going to be called sarcoplasm. So instead of cytoplasm, we call it sarcoplasm. And then you'll see this large net of purple tubing, and this is going to be called the Sarcoplasmic Reticulum, or the SR for short. And this is just going to be the muscle cell's type of smooth endoplasmic reticulum. And this is going to have a very specific function within muscle cells that you're not going to see in other types of cells. So if you look more closely at the sarcoplasmic reticulum, you can see it's going to be composed of all these tubes, or interconnected hollow tubes, and we're going to call these T-tubules. They stand for transverse tubules. And they're transverse because they travel deep within the muscle cell. So you see them here on the outside, but they're going to go all the way in so that you have this network of tubes that are able to connect to each of these fibers on the inner aspect of the muscle. And what's important about the sarcoplasmic reticulum, or the T-tubules, is that they store and release calcium ions. So we talked about how those action potentials are going to use electrical impulses to send signals, and calcium is going to be an important ion within that role. So again, we're just going to keep zooming in from here. So if you look, there's going to be all these multiple smaller tubes within each muscle cell. And it's these smaller tubes that we call myofibrils. So these are going to be muscle fibers within the muscle cell. So you can see there's many of them in the muscle cell themselves. And these are going to be the proteins that are responsible for the actual contractions of the muscle tissue. So each of these myofibrils is going to be divided into sections that run its entire length. So the sections, or these muscle units on the myofibrils, are going to be called sarcomeres. And sarcomeres are very important because they're the basic unit of contraction within the muscle. So basically, when we say a muscle contraction shortens, the physical process of contraction occurs in each of these sarcomeres. So as this one contracts here in the middle, this one contracts, and all those tiny contractions are going to build up and produce a large movement throughout the entire muscle. So the important part of this chapter is understanding what's going on in this sarcomere that allows for that shortening of these protein fibers. So each sarcomere is going to have distinct boundaries called Z-lines or Z-disc, named because when you look at them under a microscope, they're going to zigzag back and forth. So one Z-line here and one Z-line here. Everything in between is going to be the entire sarcomere. So within these myofibrils, we have even smaller proteins called myofilaments. Filaments because they're very fine fibers. And there's going to be two major types of myofilaments that we need to understand. We're going to have a thick filament, thick because it's larger. So here, the thick filament is going to encompass the central area of the sarcomere. And the protein that makes up this thick filament is going to be called myosin. If we have a thick filament, you can kind of guess that we're going to have a thin filament as well. This thin filament is going to be above and below these myosin thick filaments. And this is going to be made primarily of a protein called actin. So anytime you see the terms actin and myosin, know that we're talking about the proteins within the myofilaments. So we're going to look at how these two, basically they're going to be attracted to each other, and this myosin is going to pull this actin towards the center of the sarcomere. And because the actin is connected to the Z-line, as myosin pulls on that string of actin, the Z-lines are going to follow it to the center, and it's going to shrink or shorten that that sarcomere. So again, kind of looking at this diagram here, the thin filaments are importantly connected to these Z-lines. So if you pull on the thin filament, the Z-line follows. So looking more closely at the thin filament, we've zoomed in, and now we have it down here. There's going to be three major proteins on the thin filament that are going to be important for this process. As we already said, it's going to be mostly comprised of a protein called actin. So in this diagram, actin is going to be all these green circles going up and down the filament itself. And actin is going to serve as the binding site, so the area in which myosin is going to attach itself to, be able to grab onto it and pull it. But we don't always want myosin to be attached to, so we have other proteins to block that. So in this case, this red filament going spiraling around the actin is called tropomyosin. So like I said, tropomyosin coils around the actin, and it's going to block the binding sites on the actin to prevent myosin from inadvertently attaching itself when we don't want it to. So tropomyosin prevents myosin from binding while the muscle is at rest. So obviously our muscles aren't always contracted, so it's tropomyosin's job to keep it that way. And lastly, because we do want to contract those muscles sometimes, we're going to need another protein called troponin. And when troponin becomes activated, it's going to move tropomyosin physically out of the way and open up the binding sites on actin to allow the myosin to attach itself. And the signal that's going to tell troponin either to move tropomyosin or not is going to be calcium. So we just talked about how calcium is important in muscle cell function. So the calcium is going to leave that sarcoplasmic reticulum. It's going to attach itself to troponin, move tropomyosin, and myosin will end up binding to actin, producing a contraction. So we're going to break this whole process down much more slowly and look at it and see how that is physically accomplished. And then looking at the thick filaments, again, these are going to be located in the center of the sarcomere. They're called thick because they're much larger than the thin filaments or the actin. And this is going to be, the thick filaments are going to be mostly made of a myosin protein. So here, down here, we can see our myosin shaft, and we're going to have these heads of myosin extending out, and it's these heads that are going to attach themselves to these actin binding sites. So like we said, myosin heads attached to the actin. And what's important is that this myosin protein has a motor ability. So it means it has the ability to move. So it's going to use ATP to take that head, grab on, and physically pull it towards the center. So this action of attaching to the actin filament is going to be called cross-bridging. So it's a physical bridge formed between the thick and thin filament. Once that bridge is formed, the myosin is going to be able to pull it, and the contraction occurs. So looking more, kind of zooming back out at the larger picture, we have our motor neuron here and the connection between the muscle cells themselves. This area is going to be called the neuromuscular junction. So the area where the nervous system and the muscular system join together. So again, this is going to be called the axon of a neuron. The axon is going to extend all the way out from the cell body, which is in the spinal cord, and travel down and touch them and connect onto the muscle in which it's acting on. And again, that stimulation is going to be called the action potential. So that action potential is the electrical impulse that the nerve and muscles respond to. So zooming in at that neuromuscular junction, here we're looking at the cell membrane of the muscle, again called the sarcolemma. So everything up here is extracellular, and everything in here is going to be intracellular. So this is all inside the muscle. So inside, and this is outside. Now we've added our motor neurons, so we've joined the muscle and nervous system together. And you'll notice that it's not a direct physical connection like how two electrical wires connect. There's always, almost always going to be a space between where a nerve innervates and what it's trying to signal to. And this space is important because the way the nervous system communicates with either muscles or other nerves is it's going to release these chemical signals into this open space. And then these chemical signals called neurotransmitters are going to be able to float around and connect to the different receptors on the outside of cell membranes. So here we're going to build up a sarcomere from scratch. So in order to start, we're going to need to define our boundaries. So these boundaries again are the Z lines here on the left side and the right side. And then everything in the middle here is going to be our sarcomere. So our goal then is to bring these two Z lines closer together, physically closer together, and that's what a contraction is. So in order to bring the Z lines closer, we're going to need, bring them closer like this, we're going to need something connected to them, something to pull on to allow them. And this is the job of the thin filament. Because the thin filament is connected to the Z line, if we were to pull on it towards the center, the Z lines would automatically follow. So it's as simple as that. So now we need something to grab onto these thin filaments, and that's going to be the job of the thick filament. The thick filament is going to be what gives us that anchor or that pulling point to bring the Z lines closer together. So in order to grab onto these thin filaments, we're going to have these heads extending, these myosin heads extending from the thick filaments, and they're going to be close enough to be able to grab onto that thin filament. You'll see how they're on a slant. So once they grab on, they're going to be able to move themselves towards the center. But they can't grab onto any point on the thin filament. There's going to be specific sites on that thin filament called the myosin head binding sites. So this head myosin can only bind to this yellow area here. So in order for this to occur, we're going to need ATP. ATP is going to be the energy that the myosin head uses to do that actual pulling action. So as this ATP moves closer, connects to the myosin itself, now we can grab onto the thin filament. You can see we have this cross bridge where the thick and thin filaments are bound together. And then once it's bound, ATP comes and it's going to physically pull those Z lines closer. And this is a contraction. So it's as simple as that. It's the myosin head attaching to the actin, ATP coming in, and then pulling the sarcomere closer together. And then for these myosin heads to let go, ATP is again going to be needed for the myosin heads to relax. So ATP comes, myosin heads let go of the actin, and we have the Z lines going back to the original shape, and this is what relaxation is as well. So because we don't constantly want to be in a state of contraction, we're going to need special proteins around these actin binding sites that prevent these heads from constantly grabbing on and pulling them. So here, that's going to be the job of tropomyosin. Tropomyosin is going to be a physical blockade on these actin binding sites so that the heads of the myosin are unable to attach themselves whenever they want. But in order to eventually move this tropomyosin, because we do want to contract at certain points, the job of tropomyosin is to move tropomyosin off these binding sites. And the only way that this happens is if troponin is stimulated by calcium. So here we can see if we have free calcium ions floating around, they're going to eventually stimulate this troponin. So they're going to latch onto troponin. Once the calcium binds the troponin, it's going to physically pull tropomyosin off of the actin binding site. And now these myosin heads are going to be free to join up together. So we're going to zoom out then and look at our neuromuscular junction. Again, this is our sarcolemma out here. So this is all extracellular. This is all intracellular. So again, our cell membrane, and then these yellow areas blocking off or storing all of our calcium is the sarcoplasmic reticulum. So you can see we have a high concentration of calcium on the inside and no calcium within the T tubules. So if we were to open up this gate to the sarcoplasmic reticulum, calcium is going to naturally flow down its concentration gradient and go within the entire muscle belly or muscle cell. So in order to open these gates, we're going to need a signal from the nervous system. So we're going to add gates to this inside of the muscle. So here we have receptors that are going to be sensitive to the neurotransmitter released by the motor neuron called acetylcholine. So we've added our motor neuron now. If you notice too, we have a lot of sodium on the outside in the extracellular spaces. And when we talk about action potentials, it's going to be the flow of sodium down the neuron, and then in sodium, when it gets into the side of the muscle, the action potentials continued. So the goal of this entire situation is we want to transmit sodium from the outside to the inside. Once that sodium gets inside the muscle cell, we've transmitted our action potential, and we can have an action on these different receptor gates. So what's going to happen is our motor neuron is going to fire an action potential, and then this action potential is going to start a cascade of events which causes this neurotransmitter to be released into the synaptic cleft. So now we have acetylcholine in that synaptic cleft, and if you notice, the shape of this acetylcholine fits perfectly in this receptor binding site on the sarcolemma. So as that acetylcholine floats around, it's going to attach itself to this receptor, and once this special key fits this lock, the gate to the receptor opens, sodium flows down its concentration gradient, it's going to open this gate, and now calcium has been released. So let's kind of look at that again. The goal is we want to open up this gate. Once this gate's open, sodium comes down its concentration gradient, enter the muscle cell, and you'll notice there's this plus sign on these gates on the sarcoplasmic reticulum. So when this plus positive ion comes near this positive charge on the gate, it's going to repel it, and as that positive section of the protein's repelled, it's going to pull the gate of the door of the gate open, and that's what calcium lets out. So now we're going to open up the cell membrane receptor. Sodium goes near this positive sign, moves it away, the gate door follows it, and now calcium goes down its concentration gradient and enters the T tubules. With calcium entering the cell inside the cell, it attaches to the troponin. When the calcium attaches to the troponin, it's going to move the tropomyosin. With the tropomyosin moves, that means the myosin head can bind to the actin binding site. ATP comes on in. We've now made our cross bridge, and we're going to pull this Z lines together, and that's our contraction. ATP comes to let go of the myosin head, and we have relaxation again. So here I kind of spelled out all the individual steps. I'm going to post a YouTube video that kind of shows the animation in a little bit clearer detail than the PowerPoint might. So the big thing with learning this is to just kind of watch that video a couple times, watch this video a couple times, and read your book, and you'll slowly kind of piece together. The sequences are hard. It's a lot of the names and the terminology that makes it difficult to understand initially.